Welcome everyone to the CCPO-ICAR seminar series. It's one of my favorites in this series. Every spring semester, we host the Virginia Seagrant Fellows that we have here at ODU. We're very, very proud of the work and the research that they're doing, and we'd like to kind of highlight the work, the amazing work that they're doing. So today we have 4 Virginia Seagrant Fellows joining us, and they will be presenting their research briefly. We will hold all questions until the end of all of the presentations. And we will start with Jem, and then Alyssa, Ella, and Zlatka. So I'm going to let Jem kick us off. Welcome, Jem. Welcome, everyone. And I'm excited to hear about this research.
Thanks, Ree. Hi, everyone. Good afternoon. My name is Baldisimo. I'm currently a PhD candidate in the Ecological Sciences program at Old Dominion University. I was a Virginia Seagrant Graduate Student Fellow in 2021 until 2023, actually. But currently, I am a 2025 Canals Marine Policy Fellow working under the Coral Reef Conservation Program of NOAA. So today I want to talk about helping Nemo, communicating research on the vulnerability of fishes in the aquarium trade to promote responsible aquarium keeping. And so just to give a brief overview of the talk that I'll be giving today, I'll briefly talk about my graduate research, but go on and really focus on the outreach plan that I want to highlight this time, because over the past few years, I've been presenting about the graduate research, but I want to also highlight that something different about the Seagrant Fellowship is the outreach plan and the professional development that you actually gain from this fellowship. And so I want to talk about those two things and also some broader impacts of the graduate research that I was able to do thanks to the Virginia Seagrant Fellowship. And so the research that I did was mainly on assessing the vulnerability of marine ornamental fishes, which eventually throughout this talk, I'm going to refer to as the marine aquarium trade, or MAT. And I used the method called productivity susceptibility analysis to assess vulnerability. So this project was a collaboration with the University of Massachusetts in Boston and Roger Williams University. And essentially, this study on the aquarium trade was really done because there is a need to determine the impacts of the trade on fish populations and also identify species for research and conservation. So for Virginia itself in 2015, so this was about 10 years ago, there was actually a marine ornamental initiative which was launched. And this was a partnership between VIMS, Virginia Tech, and Virginia Seagrant. So where my project comes in is the objective of the project was to create a tool assessing the vulnerability of fishes in the trade to overfishing, and then make a list of these species that should be prioritized. So given that there is currently just mostly wild caught fishes that are in the trade, aquaculture actually provides an opportunity for us to somehow lessen the pressure on wild populations. But we don't know which species to research on or conserve if we don't really know the impact of the trade on these fish populations. So this is the thing that we were trying to address through the project. And so the method that we used could be summarized into 3 steps. So first, we looked at the characteristics of these species. We scored their productivity and susceptibility characteristics. So these are intrinsic characteristics of these fishes. And then the susceptibility pertains to something related to the trade. So is this fish, a top export or not really a top export? Is this fish widely distributed or only restricted to a certain range? Step 2 involves calculation of vulnerability. So this is based off of a NOAA toolkit that was used or that has been used for stock assessments. We used that same formula. And then for the 3rd step, we wanted to have a prioritization. So it's not enough for you to be able to calculate the vulnerability score of this fish species. If you are a resource manager, you have to know how to prioritize, because if you just have the score, you wouldn't really know what that score means. Like imagine having a list of 350 if the scores are too close to each other, how do you know which one is the least vulnerable, the moderately vulnerable, and the highly vulnerable species? And so to highlight the results of this study that we finished late last year, we did find that, as expected, some families are more susceptible to overfishing than others. We also found that there is a need for more studies on the life history of most of these fishes. So if you look at this diagram here, a lot of the purple parts per factor that we looked at represent the information that we got at the species level. So we did a lot of looking at the genus level or even the family level for these species. But lastly, despite the fact that we encountered those missing life history characteristics, we were still able to come up with a priority list of species for research and conservation that in the end, we concluded that this method that we used is actually accessible and semi-quantitative. And you can actually use it if you're a resource manager. You could use it to assess vulnerability and identify any research priorities that you would need. And so moving on to the meat of this presentation, I wanted to focus on how we actually communicated the results of our study. Given that we have this already and we came up with that list, how else could we communicate our results to the public? And here's where the outreach plan tied with my Sea Grant fellowship comes in. And so Virginia Sea Grant, just as a background, has a cross-cutting principle of knowledge integration. So it's very important that we integrate the knowledge that we find and also create strategic partnerships and collaborations. So as part of my fellowship, I had a professional mentor from the Virginia Seafood Agricultural Research Extension Center, Dr. Jonathan Van Senten. And with his help and some other people who were staff from VSRX who helped me look into other ways to make strategic partnerships and collaborations, we devised ways to create a product that we could use to package all this information. I also worked with the Virginia Aquarium through their education section or office. Ms. Karen Burns from Virginia Aquarium also gave some insight on what we could do to communicate this information to the public. So in doing so, we thought that maybe the best target audience would be for consumers and local pet shops. So with the US, the United States being a top importer of these fishes, and with consumers being the people who actually can shape the demand, it would be really beneficial to create something that can help people decide whether what they're buying as their aquarium pet is, which one would be a sustainable option. Sorry. And so our goal was to create a product that can promote sustainable decisions. And I was able to commission a friend of mine to assist in creating some illustrations for the posters. And we also asked around in the Hampton Roads area if there were any pet shops that were willing to participate. And we were able to get one, MC Aquatics. Unfortunately, they closed down, but they did provide a lot of good information that we could use for this project. And Collie Avenue Copies, so shout out to them because they also sponsored some of the products that we printed. And so this is an example of a poster that we came up with. So one of the things that I really learned throughout this whole exercise is how to package your message so that it has a good recall. A lot of people will understand it and really be able to use it if they do want to be a responsible aquarium keeper. So this is an example of the poster that we were able to print out. We made smaller versions of these. And at the back of those bookmarks, you have a description of what type of fishes are preferred if you're a newbie aquarium fish owner, what fishes you have to be wary of or cautious of purchasing, and fishes that you should avoid. And so we also made stickers, and this was really a hit for kids that we were able to meet, because they were really colorful and you could put them anywhere. And we did hold a few tabling sessions in the Virginia Aquarium for adults and kids alike to be able to know more about the ABCs of responsible aquarium keeping. Another thing that I wanted to note was that in the execution of this outreach plan, or this outreach project, we received an award from the Communication and Engagement section of the Ecological Society of America. So it was really a great opportunity for me to be able to learn how to distill, complex ideas and be able to share that to others. So moving on to the next part of my presentation, I wanted to just briefly talk also about the professional development that I was also able to get out of the fellowship. So aside from getting training in science communication and also being able to do my research itself, I was able to visit the University of Florida's Tropical Aquaculture Laboratory with the suggestion of my professional mentor. He was able to connect me with this university, and I really found out more about the aquaculture side of things. So coming into this fellowship, a lot of my experience was mostly fishery studies and the ecological aspect of marine ornamental fishing. And so this trip really enriched my understanding of the aquaculture side of marine ornamentals. I was also able to, thanks to this fellowship, attend a few other conferences where I was able to present my research and present on the outreach plan. And here you can also see my little baby who came with me to all these. So as I was growing in this career, I'm also like growing as a new mom. So for the last part of this presentation, I just wanted to highlight a few broader impacts and future directions as I've finished this research and outreach plan. The first one was that because of the project that I was able to do, I was able to contribute to information for IUCN red listing of marine ornamental fishes. There was a CITES meeting in Australia for marine ornamentals, and the research that I did with my collaborators was used for that meeting as well. So they're looking into using the method that we developed for marine ornamental fishes globally. In a country level context, I was able to also use the information that I generated to help the Philippines in making their own national red list. And I'm currently trying to publish that part of that because I'm trying to publish one chapter of my dissertation, which is relating to top 100 species in the aquarium trade that is from the Philippines. And lastly, in terms of outreach, so because I do still have all those information and pamphlets and posters, I'm able to continue the outreach even after I've already finished the Sea Grant Fellowship. And so I have an upcoming opportunity with the Smithsonian here in DC, where I'm planning to discuss the ABCs of responsible aquarium keeping.
Thank you so much, Jem. I'll let Alyssa kind of cue up her presentation.
Hi, everyone. My name is Alyssa. I am a master's student in the Ocean and Earth Sciences Department, and today I'll be talking about my research that looks at nutrients and Enterococcus bacteria in tidal floodwater. This research is made possible by the community science program Measure the Muck, which you might have seen a flyer or two about or heard me pestering you to come volunteer. And we run that program in collaboration with the Catch the King program, which is a community science tidal flood mapping program that's put on by the nonprofit Wetlands Watch. So as climate change has driven accelerating sea level rise and the eastern area, Hampton Roads area is also subsiding, we're seeing worsening tidal flooding. This graph here is showing the hours per year flooded in Norfolk from 1930 to the present. And as you can see, the trend is getting a lot worse. The past 9 years were among the top 10 most flooded years within this time period, with last year, 2024, being the worst by far. Tidal flooding poses risks to water quality in the Chesapeake Bay because when the floodwater comes up onto the land, sits there for a couple hours, and then washes back into the waterways, it carries with it a lot of pollutants, contaminants, trash, etc. So that's why we're concerned with studying the tidal floods and water quality. The constituents we're specifically looking at in this research are nutrients and bacteria that is related to fecal matter. So to study these two constituents, we need to start by understanding what's in the water before it floods. So in the baseline condition, that's corresponding to this top left panel. Then we go out and we take samples while it's flooding, and we measure the constituents in that same flood water, and then we can subtract the baseline conditions from the flooded conditions to understand how the flood is impacting water quality in our waterways. We're interested in studying nutrients because, as many of you are probably aware, nutrients are the primary pollutant that's affecting water quality in the Chesapeake Bay. The Chesapeake Bay has been experiencing impacts of nutrient pollution like eutrophication and hypoxia, or low oxygen zones, for decades. And while we have made strides in addressing nutrient pollution, and there have been reductions in wastewater and atmospheric sources, we're still continuing to have impacts. And part of the problem may be that we are not accurately characterizing all of the nutrient sources. The research we've done so far in tidal floods have shown that tidal floods are a source of nutrients to the Chesapeake Bay, but they are not being counted as a source in the regulatory documents, water quality models, or in restoration plans. And we're also interested in a different constituent, the fecal indicator bacteria, for slightly different reasons. So I said that I was studying Enterococcus. Enterococcus is a type of bacteria that is associated with fecal matter. And this includes fecal matter, or poop, from animals like dogs and birds, and also from people. This is the bacteria that's the standard for analysis in saltwater recreational areas, so beaches. So if you see a beach closure, perhaps after a storm, this is the bacteria they were testing for to determine if that beach needed to be closed. We test for this bacteria in the floodwaters, and we have unfortunately found it in high concentrations in pretty much every flood that we've sampled. This is a problem, and because people have to interact with the floodwater, even though it's not a recreational area. Clearly, as you can see from this photo on the bottom here, people do recreate in it, they let their children play in it, and they don't really have a good understanding of the risks associated with that. The Measure the Muck program and the associated water quality research have been going on since 2017. It started with a former graduate student in my lab, Alfonso Macias Tapia. And here is just a summary of what he found before I got here and started my master's degree. He did research in the Lafayette River, which is right nearby, near campus, and he found that in every flood that they sampled, dissolved nutrient concentrations were higher than the baseline concentrations in the estuary before the flood. And he found that they were so high that a single king tide flood, so that's the biggest flood of the year, could deliver more than 100% of the nitrogen that is allocated to the Lafayette River watershed through the Chesapeake Bay TMDL. So that's the whole year's worth of nitrogen delivered in a single day. That's pretty big. And that's not being counted through the TMDL. He also found that more than 80% of the floodwater samples that they tested for fecal indicator bacteria had concentrations above the recreational safe contact limit. To build on the research that he was doing, I am interested in whether we are seeing these same patterns in nutrient and Enterococcus concentrations in different watersheds, and whether there is any correlation between land use and water quality. So to answer these questions, we have expanded sampling, Measure the Muck sampling, from just the Lafayette River to 2 new watersheds, the eastern branch of the Elizabeth River, which is a primarily industrial area in downtown Norfolk, and to Gloucester Point up near where VIMS is. And that area is primarily rural or rural residential, so it's very different in land use than the Lafayette River and other Norfolk locations. Tidal flooding poses a unique challenge for sampling because it covers a huge geographic area over a really short period of time. They usually only flood for like 1 to 2 hours. And we're trying to cover even more area than we were when this project started. So to be able to make this possible, we employ community science, which you also might know as citizen science. We recruit volunteers from communities that are affected by flooding here in Norfolk and up in the Gloucester Point area. And we have them help us sample. We meet up with them in a location about an hour before the flooding starts, excuse me, and we send them out with bottles, maps, coolers, gloves, most importantly, and everybody takes samples all at the same time. So it's a big community event, and in addition to being beneficial to our research by providing data, it also benefits the community by providing environmental education and giving people firsthand experience doing science. So just as an example of how this program has impacted the people who volunteered, I have a short video that was sent to me by the Girl Scouts who participated in Measure the Muck last fall. So it's really fun getting to work with all of the community members who volunteer for this event. Here's just a few more pictures of some of the few people that have volunteered, a few of the many people, I'm sorry, who have volunteered for us over the years. And you can see in the kind of the bottom left here, all of us meeting up and distributing coolers. So this is really a highlight of the work that I get to do. So now I'd like to pivot to a little more of the science heavy side, just to show some initial results of what we've found since I started. I started my master's degree in fall of 2023, and since then we have conducted 5 community science sampling events, those Measure the Muck events. And here, this shows some initial data from the fall 2023 and spring 2024 events. So you'll see there's 2 different watersheds represented here. On the left, there's data from the eastern branch of the Elizabeth River, that's in downtown Norfolk, and on the right from the Lafayette River, which is right here near campus. And these plots are showing dissolved inorganic nitrogen and dissolved inorganic phosphorus, so 2 different forms of dissolved nutrients in each of these watersheds. Within the plots, the box plots represent the range of floodwater concentrations, and the blue and orange dots represent baseline conditions before the flooding happened. So the big takeaway from these graphs is that in every instance, floodwater concentrations are higher than baseline. So this shows a similar pattern to what Alfonso found in his research before me, that floods are a source of nutrients to both of these waterways. Another key thing to take away from these plots is that there's really high variability in the concentrations of nutrients in the floodwater. So for example, in this plot of dissolved inorganic phosphorus on the left here, you can see in September, the concentrations ranged from essentially 0 all the way up to over 20 micromolar. So it's important to keep this in mind as we're thinking about how to analyze the data and how land use is interacting with nutrients. And so here are some similar plots of Enterococcus concentrations during those same flood events. And again, we're seeing similar patterns to nutrients. We're seeing that the floods have consistently higher concentrations than the baseline, and there again is really high variability in the concentrations within the floodwater. And then I'd like to close with some initial data that came from last fall's sampling events. So we had 2 Measure the Muck community science events in September and October corresponding with the king tides. In September, we covered the Lafayette River, which is on the top here, and the eastern branch of the Elizabeth River in Norfolk. We had 47 volunteers who collected a record 282 samples. This was the most samples that's ever been collected through a Measure the Muck event. It was also the worst flooding Norfolk had experienced since 1930, so that kind of makes sense. While we're still working on getting the nutrient data back, we found that 82% of the samples we collected during that flood had Enterococcus bacteria concentrations over the safe contact limit. So that's, I think, a really big deal and something we should be talking about more and spreading more awareness about to the people whose communities are being flooded. Here are a selection of photos of the flooding that were submitted by volunteers. When they're out there collecting samples, they're also taking photos, GPS points, and recording information about their environment, like what is the land use and where is the floodwater coming from. In October, we sampled some of those same areas in Norfolk, and we also expanded sampling up to the Gloucester Point area for the first time. So I'm really interested to see how the nutrients and bacteria in those rural areas compared to the urban samples that we're getting from Norfolk. And here are some photos highlighting just how different that Gloucester Point area is from Norfolk. The flooded areas here have a lot more wetland ecosystems, gravel roads, and like neighborhoods that are just significantly less urban than Norfolk is. That's all I have for you today. I'd like to close by just thanking our funders and partners. We definitely could not do it without them.
Thank you so much, Alyssa. I'll let Ella queue up for her presentation. Ella, go ahead.
Good afternoon, everyone. My name is Ella DiPetto. I'm a third-year PhD candidate at Old Dominion University. I work in Dr. Eric Walters' lab, and I'm also a Virginia Sea Grant Fellow. I'm excited to be here, so thanks for having me. I'm going to give an update on a recent project that's supported in part by Virginia Sea Grant and my professional mentor, Dr. Wee Youssef. So thank you, Wee, a professor in the School of Public Service here at ODU. This project aims to enhance the regional knowledge transfer surrounding living shoreline implementation. So essentially trying to connect and elevate those involved in on-the-ground living shoreline work at a regional scale. My dissertation work is focused on living shorelines, which are a newer shoreline protection technique that emphasizes natural materials to stabilize shorelines. There's a growing interest in living shorelines because of their ecological benefits compared to traditional hard shoreline protection measures like seawalls or bulkheads, but also because of their potential to adapt with climate change. Here in Hampton Roads, at a typical living shoreline design like the one in this photo, we typically fill an eroding shoreline with sand, plant marsh vegetation, and often add an oyster sill, like the oyster castles towards the front of this picture, to further aid in stabilization, but also provide a substrate for oysters. The term living shoreline encompasses a huge variety of different designs, construction materials, composition, and project goals. All of these pictures here are considered living shorelines, from proprietary mixtures of concrete and shell, like the natrix or oyster castles, to rock sills, and even bags of oyster shell. And all of this complexity and variability makes design standards and solidifying best practices really hard to develop. So there's a lot of small scale knowledge being produced by the practitioners who are designing and building these shorelines, but this knowledge can often remain siloed or separate from one another, staying within an organization or a locality, and remains widely unknown to other practitioners building similar types of living shorelines. So this has led me to become interested in how living shoreline knowledge was being transferred, shared, and what kind of knowledge was being exchanged. So I narrowed my focus to the eastern and Gulf Coast of the US, which have relatively similar ecological conditions and coastal species, but still exhibit a lot of variability. And the objectives of this research project are to synthesize living shoreline implementation best practices. How are the folks who do this every day, how are they approaching some of these challenges? Understand the state of practices. So again, identifying implementation challenges, opportunities, but also trying to solicit some of these novel solutions that some of these folks have come up with. Also understand the state of knowledge exchange. How are these folks sharing knowledge amongst themselves and amongst other groups? And I also wanted to connect practitioners to one another at a regional scale. I'm achieving these objectives through a combination of surveys, interviews, and collaborative workshops, and I'll walk you through my research methods, giving updates on work I've already conducted and orienting everyone to my next steps. In the fall of 2024, I created and sent out an online questionnaire to gather initial background and demographic data of practitioners. Participants included those involved in the design, permitting, or construction of living shorelines within the Gulf or East Coast states. Overall, at the end of this intake survey, 18 practitioners agreed to participate in the project, and data from the intake survey showed a majority of these individuals worked in the nonprofit and private sector, with 10 to 7 respectively, and 1 from a government agency or the public sector. In terms of organization size, where these individuals were working, it was well distributed, ranged from really small organizations with less than 10 employees. Usually these were at the local or sub-local scales, but also ranged to larger organizations with over 100 employees. These practitioners worked at different scales, with some only working, again, in this like sub-watershed, very local scale, others working within a state, and others working across multiple states or even internationally. So in total, 15 states were represented, with work in Virginia and Florida being the most common. You can see those darker blue colorations on this graph right here. So overall, there was a lot of variety in the type of groups, the scope of work, and the geography. Following the intake survey, I don't know why I keep saying intake surgery. I independently met with and interviewed each of these practitioners over Zoom. And these conversations lasted from about an hour to 90 minutes. And I followed the semi-structured interview process, which means I had a lot of freedom to follow up and encourage discussion with all of these participants. I was able to dig deeper into experiences and worldviews regarding these 2 major concepts. So best practices and lessons learned, and also knowledge acquisition and sharing. So currently, I'm analyzing the information from all of these interviews, pulling together common themes across these 2 sections. This analysis is ongoing, but I wanted to highlight some of the recurring concepts I'm starting to see across all of these interviews. So practitioner collaboration with academic researchers to answer questions. That's been a big thing that's come up. Also, trying to figure out innovative ways to increase planted vegetation success. We're seeing a lot of challenges with getting a lot of the marsh species to establish, and this isn't only in Virginia, this is across the entire range of those participants. There's also a desire for guidance on how to incorporate climate change and sea level rise into living shoreline design. And there's a huge emphasis with learning from trial and error. And of course, lots of regulatory hurdles in getting these projects permitted, designed, and constructed from across the geographic scope. So thinking about accessible ways to share and build upon this information with practitioners, one of the outputs of this project will be a collaborative workshop with practitioners. I am planning to host a session at this fall's Restore America's Estuaries Living Shorelines Tech Transfer Workshop. This is an event that aims to connect those working on living shorelines across the country, often policymakers, regulators, marine contractors, and nonprofits, those involved, again, in that on-the-ground work and less so on that research and academic side. I'll be inviting all of the project participants and opening up the session to a wider practitioner audience to try and get even more of those experiences shared. And I'm really excited about this in-person opportunity to dig into the emerging concepts and the challenges and work on identifying practically derived solutions to a lot of these challenges. Finally, my last step will be to produce a technical document with practitioners as the intended audience. I think a lot of the time we as researchers, we write academic papers and we present at academic conferences to other researchers, but we aren't always reaching the groups we want to reach with or using methods of education and sharing that are accessible to them. So I think like a lot of the fellows here today, and this is a really big emphasis of Sea Grant, is kind of reaching your audience in new and accessible ways. So it's really important to me that this project remains practitioner focused from start to finish. Increasing the availability of this practically gained knowledge, best practices, and innovative solutions to a lot of the emerging challenges in coastal protection is needed to ensure we're maximizing the benefits of living shorelines so that they're protecting societies better for longer and also They are restoring ecosystems. And so through this project, I hope to elevate this knowledge and advance the effectiveness of living shorelines within Virginia, but also across the country. Thank you for your time. I look forward to sharing more of my results with you all in the future. This was built upon the proposal of the project at last one of these seminars. So it's really been nice to kind of have this iterative approach to sharing with you all the progress on this project. I want to acknowledge Virginia Sea Grant specifically and the support they give beyond research, but in education and professional development. It's been absolutely wonderful to work with them. It's always been a priority of mine to integrate people with nature, and they've supported me in finding the tools to do that. I'd also like to thank my dissertation committee, my research lab, and all of the other forms of support for this project. So thank you very much.
Thank you, Ella. And we'll let Zlatka finish us off with the presentations. Go ahead.
Thank you so much for this opportunity. I am Zlatka Rebolledo Sanchez, also a PhD candidate in the Ecological Sciences program here at ODU. And today I want to talk about how I am translating science to general audiences, using as an example what I do for my research, but this is supposed to be used for any others. If you're working in even, I don't know, computer science or other systems, if you want to engage with different audiences, you should be able to use like this similar approach. So first, a little bit about my research. I work with blue carbon in salt marshes. So blue carbon is a ecosystems that sequester and stored a lot of carbon in them, like seagrasses, salt marshes, and mangroves. And they're currently used for climate change and mitigation adaptation, if just focusing on their preservation, restoring them, and utilizing them, because they're amazing doing these ecosystem services. But we need to properly communicate this information, and we need to properly estimate this amount of, to have realistic like targets. Typical studies for blue carbon, it's just take one or just a few cores in one site, and then they estimate the whole area of the site, and they account like all the patches are the same and there's no difference, and then they come up with one single score. So from my previous results, I see there are differences across different sites, but also inside of each site, we have difference between patches depending on their size and their shape. And then inside of the patches, we have also differences. So I wanted to dig in a little bit more on that inside the patches, and I realized that this is an example in the left side, that usually it's just that one value, and that will be the estimated value of one of my sites if we only have that estimation from one core. But then we have way more variability in these pools, in the above ground, below ground, and in the soil. So yeah, it's way more heterogeneous than that usually the science expects. So why this matters? Well, it's very important for all the carbon credit market. A lot of people now, it's using these ecosystems to generate credits and for profit. So to properly estimate this is very important. Also, the way we are sharing the data and the data that we are collecting, it needs to be improved. There is currently like a national and international effort to include more soil carbon data and share it with others. However, for the state of Virginia, we have really low quality data currently, and we don't have much of the coverage of all our salt marshes. And then in the world, we need to be able to compare to other sites and see if this applies to other sites. And to do all these different things, we need to properly communicate with different teams. So have effective communication of our science. And this will look like different depending on who you are engaging. So this is the approach that I created. First, I focus on the audience, then the specific goals development, how I'm going to execute and engage with the different group, and then integrate the feedback from that group. So first, we need to be really, really specific and know your audience that you're engaging. Try to come up with specific ways to connect and find common ground. This is a image, something that Alyssa was mentioning in our title. floods in the area, this is a great way to start. This is something that it's very annoying in this area, and we can talk to someone in the street just about this. Like it's very annoying. We hope to change this. And one way is having more living shorelands or using these salt marshes and other, and then we can talk about other ecosystem services, not just the flooding prevention or the erosion control. We can talk about carbon sequestration and other things that are related. Here are a couple of pictures of other outreach events that I have done. In these couple of years, I have been engaging with more than 3,000 participants that I have been talking about salt marshes and why they're really cool. And this is a small model of like wave attenuation, and they can choose where they build their houses. So it's like a really nice thing that we can connect with people. And obviously it's completely different if we're talking with kids compared if we are talking with their parents, or if we're talking with a nonprofit, or if we're talking in a scientific conference. So try to be as specific as possible with that audience. Then be very intentional about accessibility and representation, trying to be outside our comfort zone, so consume different social media, get exposed to different perspectives, and ask them like what are their interests, and find this common ground between them. And another thing about accessibility is trying to be intentional. And one of the language accessibilities that is what I'm focusing on. So all my outreach posters and flyers are in both English and Spanish, and I try to use not only a lot of text, but like a lot of images and things that are easy to engage with, and it's easy to understand the processes. Then, Then when we're working on our specific goals, we try to have SMARTY goals. They should be specific, measurable, attainable. They need to be relevant for our audience. Also, it should be time-bound, inclusive, and equitable. So when we are engaging with this specific audience, why is it relevant to them? What they need to know specifically. They don't need to know the specific details of my research or a specific, I don't know, carbon cycle. Only share the specific things that they are interested in. And if they have more questions, we can share more. And then specifically, why do I want to engage with them? Why this is relevant to myself? Why is it relevant to my science? And what are the reasons of this specific conversation that we're having? Then when we have that figured out, first, when we're engaging and executing our projects, we need to avoid biases and assumptions. Sometimes a lot of scientists assume like the people don't know anything, and they start conversations with an approach that it's not great for having these interactions. So we need to try to avoid this. Then we need to bring our work to our specific audience, as we mentioned before, avoid jargon. And this is like a statement that it's very simple for a scientific audience. Low carbon ecosystems like salt marshes play a vital role in mitigating climate change by sequestering and storing significant amounts of carbon dioxide, helping to reduce atmospheric greenhouse gases. But if we say this to a little kid, it's going to be a little bit confusing. So we want to divide the different things that we are saying in that statement and explain them what it's a marsh, why marshes help us protect from flooding, a little bit of what the climate emergency and climate change, or using other words that are related. We are still talking about it, but maybe we're avoiding that specific term. Why plants are important, what is photosynthesis, and why marshes are specifically special for all these CO2 sequestration and storage. Then that we need to protect these coastal wetlands, and we need to do more. So they will be more engaged, and they will be asking questions, and it's like a great way to divide this information. Then if we're talking with policymakers, well, first, why wetlands are important and all the ecosystem services, but also for the carbon credits, the economic benefits, and the social benefits that will be like used from this. So it is also dividing these different concepts, but being really, really intentional. And depending on the policymaker, maybe they are more interested in one area than others. So just to pitch that in a different way. Then this is an example of a poster that me and my intern from Virginia Sea Grant did last year. So it's different concepts of low carbon, and we have these little bubbles. So the animals are talking and the plants are giving information, and it's like an opportunity to engage with people. And we really had a lot of good feedback from this poster. And yeah, this is just one example, but there are many different ways to do this approach. Then the perception, we need to work on the perception that the public are going to take from us. So if they trust us or not, if we're if it's if we have good integrity, and how we build that trust with them. Then be very open and warm and inspire them and be excited about our projects. If we are having fun in the field, if we're having fun while we're showing them these results and how it's going to be impacted in their life, probably they're going to be way more open to hear what we want to say and what we want to share. And always try to work in having really clear what is the main takeaway, what is your general message, and try to reiterate in that. And then our last part is integrated the feedback. Always hear different perspectives, request that specific opinion from them, and be actively listening to what are their thoughts, what do they need, specific questions, and we need to adapt. So this is like a cycle, but it's going to be change. It's going to reiterate. So from that feedback, we might change our execution and engagement, and we might change even our general goal to adapt to what they actually need. We need to review everything constantly. And these are just a few strategies for effective communication that works for me. And my main takeaway for you today is that you always need to target everything to your specific audience and work with them and build with them this specific program that is going to be helpful for them. And I want to thank all my collaborators and funders, everyone in my labs and in the ODU program, other grad students in other programs, especially Virginia Sea Grant, obviously, for this amazing opportunity to be a fellow for these 2 years, and all the other funding sources as well. And if you have any questions, you can ask me via social media or email. And there's my website. So as well in the website, you can find all the different outreach events and projects that I have been doing so far. So thank you so much.